welcome to the 9th module in temperature stress physiology. Today we'll be discussing chilling, the second form of temperature stress. We will first start by defining low temperature stress and comparing chilling and freezing, and then we'll go over temperate and chilling sensitive plants and see the differences between them. Following that, we will discuss chilling injuries, both molecular and physiological. And then we'll discuss molecular injuries in more details, under which we explore more about activated and radical oxygen species and how chilling stress affects oxidative stress. And finally, we'll go back and have a look at the symptoms and explain them based on the molecular injuries we've learned. Before we start learning about chilling stress, it is important to distinguish the difference between chilling and freezing stress. Both are cases where plants are subjected to suboptimal temperatures, which can alter many plant physiological processes. The nature of stress experienced by plants exposed to cold temperature is dependent on the state of water inside the cells at that specific temperature. Therefore, low temperature stress can be classified into chilling, where stress is caused by temperatures between 0 to 15 degrees Celsius, where the water is still in its fluid state, and freezing, where stress is caused by ice crystals forming in the cell. Most higher plants living in temperate regions can tolerate low temperature stress through cold acclimation when temperatures drop down in fall and winter. However, plants living in tropical and subtropical regions show physical and physiological injuries when temperatures drop to 0 to 15 Celsius. The general consequences of chilling injury include limited geographic distribution, both latitudinally and based on elevation, limited available growing seasons for many cultivated crops. We can't grow slowly maturing crops that are chilling susceptible in areas with short growing seasons. And the third consequence would be the economic losses and the reduction in quality that are based on yield reduction and post-harvest losses that plants can suffer. Examples of susceptible crops include many plants of tropical or subtropical origin, some with C4 pathways of photosynthesis. Some of these are bananas, citrus, tomato, rice, cotton, corn, and soybean. In the upper image on the right, you can see the chilling stress effect on bananas, or what we see over here is called fruit pitting. On the bottom right, you can see the effect of chilling on ripening tomatoes. Chilling can have an irreversible damage on tomatoes and prevent them from ripening, which would be a very big economical loss. Injuries that chilling sensitive plants can suffer from can be classified into molecular and physiological injuries, or we can look at them as primary events and secondary events. As the name suggests, the secondary events will take place after the primary events happen. Primary injuries are temperature dependent, with the threshold around 10 to 15 degrees Celsius. They can lead to acclimation responses over time or to a series of secondary events which can give rise to symptoms. Let's start with the secondary events, which are the symptoms that we see when chilling sensitive plants get injured. And after that, we will look at the primary events that would explain how those symptoms affect the plant's tissue. Secondary events include the physiological changes that lead to symptoms. Examples of injury symptoms include stunted plants with reduced vigor and reduced meristematic development, altered metabolism, where we notice increased rates of CO2 and ethylene production, increased levels of anaerobic metabolites, and reduced photosynthetic rates. Male reproductive organs are usually more temperature sensitive than female organs, with pollen maturation being a weak link. Male sterility can be observed in crops such as sorghum when night temperatures are 13 degrees Celsius or less during meiosis. The problem may be related to inadequate energy resources or to depolarization of microtubules. Ripening abnormalities can occur when fruits are harvested immature and refrigerated to facilitate long-distance shipping and storage. Most of us are familiar with winter tomatoes that fail to ripen normally and develop to the full flavor and aroma of garden tomatoes. Surface lesions include pitting, as in the grapefruit in the figure. The sunken, discolored areas result from water loss from subsurface cells. Increased susceptibility to decay. When fruits or vegetables are removed from extended refrigeration, they often senesce and decay rapidly. The increased susceptibility to decay may be related to the leakage of nutrients from cells with membrane dysfunctions. The ultimate symptoms of chilling injury is loss of viability. In the next section, we will examine the molecular injuries or the primary events in more details. There are several candidates for primary events. Those include cold labile proteins, phase separations of membrane lipids, chill-induced cytoplasmic acidification, photostasis, and cytoskeleton depolymerization. They are not mutually exclusive, so more than one event can occur in a chilled plant. We will go over each one in details next. Low temperature, just like high temperatures, can affect stability of some proteins. The vacuolar V-type ATPase is an example of a temperature labile protein that loses its stability at temperatures below 12 degrees Celsius. As you remember from the membranes module, the V-type ATPase proton pump hydrolyzes ATP to pump protons into the vacuole. The V-type ATPase enzyme complex is made of two domains: a V1 peripheral domain that is responsible for the ATP hydrolysis, and a membrane integrated domain V0 that is responsible for proton pumping. Research showed that at low temperatures, the V1 domain tends to detach from the complex, causing its deactivation. The structural change causes the pump to stop working, and this decreases the enzyme activities associated with mitochondria, ER, and plasma membrane. Focusing on reactions within a pathway, we expect to observe decreased enzyme activity at lower temperatures based on kinetics. In other words, the low temperature can affect enzyme reaction rates due to limited collision. Imbalances can arise when reactions have differences in free energy of activation. Reactions with larger energies of activation are inhibited to a greater extent by low temperature. The result, as depicted in the Arrhenius plot, is that relative reaction rates can change with temperature, leading to a crossover of control. Metabolism shifts from balanced to an unbalanced situation. The second primary event or molecular injury is the phase separations of membrane lipids. Phase separation of membrane lipids can represent a temperature-dependent event that can lead to acclimation or injury. The diagram of the output from a calorimeter shows the enthalpic changes that occur when membrane lipids undergo a change in state. The phase separation, as membrane lipids go from fluid-liquid crystalline state L-alpha at ambient temperatures to a solid gel L-beta at chilling temperatures, can trigger signal transduction pathways leading to acclimation or result in metabolic lesions. The figure shows a membrane that has undergone a lateral phase separation, as would occur at chilling temperatures. The solid gel region is tightly packed and has excluded proteins which have become concentrated in the liquid crystalline domain. The original hypothesis involved a phase transition in which the entire membrane solidified, disrupting the activity of membrane-associated proteins. There have been many experiments conducted over the years, some supporting and others refuting this hypothesis. Some supporting evidence comes from freeze-fracture electron microscopy, a technique that can provide information on the disruption of membrane particles. In this micrograph of a control membrane on the left, the particles are evenly distributed. However, in the micrograph on the right, the membrane was exposed to chilling. The particles are grouped into a crescent, presumably pushed away from the solid gel portion and partitioning into the liquid crystalline area. When low temperature is sensed by the cell membrane, the lipid membrane phase separates and proton pumps are disrupted, which leads to proton effusion from the vacuole to the cytoplasm. With the absence of functional proton gradient between vacuole and cytoplasm, active secondary transport is also affected and ion homeostasis is impaired. This imbalance of ion fluxes causes the cytoplasm to acidify and metabolic systems become perturbed. If this situation is continued for prolonged periods, damage becomes irreversible and ultimately kills the cell. The fourth event that affects the molecular structure of the cell is cytoskeleton depolymerization. The cytoskeleton is another potential target for temperature-induced changes in structure. The cytoskeleton is a network of filamentous polymers of different sizes and composition. It can be made of different proteins with alpha and beta tubulin forming microtubules, while actin polymers form actin filaments. The protein units are held together by numerous non-covalent bonds. Polymer assembly and disassembly is controlled by various factors, including ionic strength and temperature. Chilling temperatures may lead to depolymerization of cytoskeleton components, which some say constitutes a primary chilling event. The second mechanism by which cytoskeleton structure can affect the cell is by looking at the spatial organization of the organelles. It is easy to visualize the far-reaching effects of changes in temperature on the cytoskeleton fibers because one function of the cytoskeleton is to maintain spatial organization by anchoring macromolecules and organelles. A low temperature-induced depolymerization of cytoskeletal fibers could be associated with breakdown of polysomes and changes in patterns of translation. Lastly, the cytoskeletal changes can also affect the function of signal transduction pathways by altering transduction clustering. The fifth primary event or molecular injury is photostasis, which is also referred to as light-dependent chilling injury. Injuries in chilling-sensitive plants have been reported to be very different in light and dark conditions. In greening plants, light has shown to deteriorate the chilling injury far more than in dark. This is thought to be due to the photooxidation of chloroplasts at low temperatures. As we discussed earlier, low temperature has a negative effect on proteins or enzyme stability, as well as on their reactions rate. The resulting temperature dependency of enzymes' activity will reduce metabolism. This will affect different enzymes in metabolic cycles, cascades, and redox chains. Chilling during light hours leads to imbalanced photostasis. Photostasis involves the balance between the energy-absorbing reactions and the carbon reduction reactions of photosynthesis. To better understand this concept, we will have a look at how activated oxygen affects photosynthesis. We talked about oxidative stress and light stress briefly in our plant stress module. Here we will go into the details of these stresses' development and how chilling is related to them. Activated oxygen molecules are referred to as activated oxygen species or reactive oxygen species. As the name implies, they are more reactive than oxygen molecules we breathe from our atmosphere. Activated oxygen in biological systems includes radical and non-radical forms. Free radicals are familiar in human biology. Anti-aging creams contain antioxidants like vitamin E. Some forms of cancer and arterial diseases involve radicals. and our bodies can fight some infectious agents using activated forms of oxygen. Activated oxygen species roles in plants has been seen in attacks on pathogens, in cell wall polymerization, and in signal transduction, as in our earlier example of hydrogen peroxide. It is fair to ask whether activated oxygen species are bad or good. To answer this, we can draw an analogy with fire. It can be very powerful when controlled, but destructive when it goes out of control. As our starting point, we will begin with oxygen, the molecule we breathe. It is also referred to as triplet ground state oxygen. It has triplet unpaired electrons with the same spin. This is the ground state and it has the lowest energy. In the case where triplet ground state oxygen accepts energy, this flips the spin of one of the electrons, forming singlet oxygen, the first species of activated oxygen. This can occur in chloroplasts when photons excite chlorophyll from the ground state to excited singlet state. Normally, chlorophyll passes along the energy to an acceptor during the photosynthetic process. If photosynthetic electron transport is impaired, the energy can be transferred to a triplet oxygen instead, forming singlet oxygens. The other way to increase reactivity is through reduction. Adding an electron to the triplet ground state forms superoxide, the second species of active oxygen. Continuing in this reduction series, adding another electron yields hydrogen peroxide. It is found in the protonated form at physiological conditions. Hydrogen peroxide is not radical since it has two sets of paired electrons. It is one of the most stable activated oxygen species, having relatively low reactivity towards most organic molecules. Hydrogen peroxide is neutral, allowing it to cross cell membranes. Upon adding another electron in the form of hydrogen atom and splitting out water, the hydroxyl radical is formed. The hydroxyl radical is extremely reactive and is one of the most potent oxidizing agents. It reacts at a diffusion limited rate, usually with the first biomolecule it contacts. Its action can be described as random destruction. Chilling-induced increases in the rate of formation of oxidative stress species in stressed tissues can either lead to acclimation or injury. Cells have a baseline level with activated oxygen constantly formed and detoxified. A small increase can be advantageous, acting as a secondary messenger and triggering the induction of stress response genes. However, a large increase can tip the scale in favor of damaging oxidative reactions. The figure is from a chilling study with maize seedlings showing increased production of activated oxygen species associated with acclimation or injury, depending on prior conditions. Scientists measured activity of hydrogen peroxide detoxifying enzymes on the left y-axis, which are CAT3, mitochondrial catalase isozyme, and POX, which is peroxidase. Hydrogen peroxide levels are reflected on the y-axis. 4 treatments were imposed in the dark. The first one was controls at 27 degrees Celsius. The second were acclimated at 14 degrees Celsius. And the third are acclimated at 14 degrees Celsius followed by 4 degrees Celsius. And the last group was controls at 27 degrees Celsius transferred directly to 4 degrees Celsius. The control non-acclimated plants placed directly into the chilling regime in group 4 had a large increase in hydrogen peroxide that was associated with injury. The acclimation treatment exhibited a small increase in hydrogen peroxide and increased defense enzyme activities, especially CAT3. The increase in hydrogen peroxide for the acclimated plants exposed to 4 degrees Celsius was substantially less than the non-acclimated treatment, presumably due to the increased enzymatic activity. If you're interested to learn more about this experiment, you can find the article in the supplementary material. Scientists generally agree that chilling stress can increase activated oxygen, with electron transport chains contributing. Mitochondria and chloroplasts contain two of the key pathways where oxygen can be reduced. In mitochondrial electron transport, the pathways are the cytochrome pathway, which produces ATP, and the alternate cyanide-resistant pathway, which produces heat. The mitochondrial electron transport is diagrammed with electron routes shown in red and proton pumping in blue. There is more than one route of mitochondrial electron transport, with the normal cytochrome pathway involving several protein complexes. In this process, reduced compounds such as NADH transfer electrons to complex 1, a large dehydrogenase. The electrons are passed through a series of intermediates, ultimately residing to O2. As the reducing potential decreases, the energy is conserved by pumping protons across the membrane. The resultant proton motive force drives phosphorylation of ADP to ATP by the F-type proton ATPase. There are additional pathways involving alternate reductases to accept electrons from reduced compounds and an alternate oxidase to reduce oxygen. Chilling interferes with normal mitochondrial electron transport through changes in membrane viscosity. The over-reduced state leads to partial reduction of triplet oxygen to activated forms instead of to water, as well as disrupting the respiratory balance and leading to a buildup of fermentation compounds. Chloroplasts also contain electron transport chains that can produce activated oxygen species in chill tissues. When chlorophyll accepts slight energy, de-excitation to the ground state can occur by emission of light at slightly different wavelengths, evolution of heat, or by reacting with other molecules. Normally, energy is passed to reaction centers via resonance transfer, leading to charge separation and generation of ATP and reducing power. However, excited chlorophyll can also react directly with oxygen, forming singlet oxygen. The lowered rate of carbon fixation at chilling temperatures disrupts the balance between energy harvesting and carbon reduction reactions. Chilling has a greater inhibitory effect on the carbon dioxide fixing enzymes and can interfere with translocation of sugars, leading to feedback inhibition. The result is an increase in ADPH to NADP plus ratio since chlorophyll is oxidized at a greater rate than NADPH is oxidized. The shortage of NADP, the normal electron acceptor, leads to over-energized reaction centers. Energy can be transferred to a triplet oxygen forming singlet oxygen, and electrons can be passed forming superoxides. Activated oxygen species can damage the reaction centers, especially the susceptible D1 protein, which is one of the scaffold proteins on photosystem II. Photoinhibition, which we defined earlier as a decline in photosynthetic rate, is a result of several processes, including defense and repair mechanisms. Chilling stress degrades the D1 protein at the reaction center much faster than normal. Repair involves a partial diassembly of PS2 to remove and break down the damaged protein and replace it with a new D1 protein. Electron transport can be further impaired by the compromised status of PS2 during repair of the reaction center. Severe chilling stress can go beyond photoinhibition, leading to chlorophyll breakdown and peroxidation of chloroplast lipids. The high unsaturated membranes are susceptible to activated oxygen species attack and subsequent chain reactions. Those will lead to reduced photosynthetic capacity, pigment degradation, degradation of chloroplast lipids. In this table, we can summarize the molecular chilling injuries in sensitive plants. The first injury affects the proteins in the vacuolar membrane. The V1 domain in the V-type ATPase protein detaches from the complex, resulting in its deactivation, with the protons effluxing to the cytoplasm due to the deactivation of the proton pumps. Cytoplasmic acidification occurs. In the thylakoid membrane, we can see that enzymes have a lower activity at lower temperatures. This would increase the concentration of activated oxygen species and eventually cause a damage in the PS1 system. Another injury that takes place at the thylakoid membrane is lipid phase separation, which affects membrane's vital components such as the electron transfer chain. This decreases the D1 turnover and damages the PS2 system. And finally, the last molecular injury that we discussed today hits the cytoskeleton and causes depolymerization of the cytoskeletal components. Now let's have a look at how molecular injuries can explain the physiological symptoms. Chilling stress can cause secondary water stress. Causes of secondary water stress involve changes in resistance to water movement. One side affected by this phenomena is the stomata, which are unable to respond via normal signal transduction pathways to reduce turgor. The regulation of gas exchange becomes very slow due to changes in the membrane's lipid state. Or in some cases, the stomatas are stuck and unable to close at chilling temperatures. If the chilling persists long enough for the roots to cool, which is more common in potted plants, they can be another site of altered water relations. Chilling can decrease hydraulic conductivity through contraction of the membrane from greater order of lipid molecules, leading to restricted movement of small molecules through the membrane. Decreased permeability can also reflect changes in aquaporine activity. Evidence suggests that resistant species have greater aquaporine activity, although other factors are likely to be also involved in membrane dysfunction in susceptible species. Taken together, plants cannot take up sufficient water when its loss is uncontrolled, leading to loss of turgor. In chilling sensitive fruits such as citrus and banana, pitting appears on the surface of the fruits. The pitting results from injury and collapse of subsurface cells, followed rapidly by invasion of decay organisms. Such visible pitting can be related to rate of water loss in some commodities. Membrane dysfunctions have been implicated in a number of chilling stress symptoms. Tissues can appear water-soaked from the leakage of cellular contents from loss of membrane semipermeability. These problems may be related to entropic changes or impairment of transport proteins. Both sensitive and resistant plants undergo membrane depolarization upon exposure to chilling temperatures, but resistant plants are better able to reestablish the electrical potential. Water soaking is readily apparent in leaves when the intracellular air spaces become filled with cellular solution. In some cases, the solution returns to the cytoplasm after stress. In others, the water-soaked areas become necrotic. And here are the questions that you should have learned the answers for in this module. What is the difference between chilling and freezing? What is the difference between temperate and chilling sensitive plants? And what are the general consequences of this difference? What is the difference between primary and secondary events? You should be able to name some secondary events and name and explain the primary events. What are the normal roles of activated oxygen species in plants? How does chilling induce activated oxygen species formation? And what are the damages that activated oxygen species can cause in plant? And finally, explain the physiological damage based on the molecular injuries. That's all for today. We'll see you in the next module.